Hello everyone. So today I will start with Unit 5 of NET syllabus, which is the developmental biology portion. মানে যেটাকে আমরা খুব commonly dev bio বলে থাকি. So এই ইউনিটটার মধ্যে আছে 5 sections. So the first section is basic concepts of development, and then moving on with the gametogenesis, fertilization, and then morphogenesis, organogenesis in plants as well as animals, and lastly the programmed cell death, aging, and senescence. So I will start with basic concepts of development in this presentation. So to start with it, I will move on with fertilization. So আমরা already জানি যে sperm and egg, which is the male gamete and the female gamete. These are haploid cells, which means they have a single set of chromosomes in them. So these two cells fuse together and they form the diploid cell, which is known as zygote. So zygote is basically the first formed diploid cell of any multicellular organism in development. So from zygote, what happens is that a series of mitotic divisions start. Now this division results in a multicellular organism. And what happens is that from the fertilized egg, which means the zygote, the 1 cell stage becomes 2 cell stage, and from here 4 cell stage, 8 cell stage, and when it reaches this 16 to 32 cell stage, this structure is commonly known as morula, which is also called a solid ball of cells. So as you can see, this entire structure is filled with cells and there is no space or hollow empty gap between. So from here, this cell stage moves on to the 100 cell stage. Like around when it reaches around 100 cells, what happens is that it forms a structure known as blastula or blastocyst. What is the speciality of this structure? The cells get arranged themselves at the boundary of the ball. So that is why it is known as a hollow ball. So from solid ball, it goes on to form a hollow ball structure, leaving a vast space within them. So this space is known as blastocele. Okay, this is a fluid filled space, but this space is formed by when the cells arrange themselves around the periphery of the ball. So from this 100 cell stage, what happens is that an invagination starts. Cells start invaginating inside and they form 3 germ layers. That means they get arranged, they arrange themselves in 3 subsequent layers, and this structure is termed as gastrula. So the process of blastula formation is known as blastulation, and the process of gastrula formation is known as gastrulation. So what are these germ layers? So these 3 germ layers are basically ectoderm, mesoderm, and endoderm. Ectoderm that is responsible for the formation of nervous system as well as the skin in later stages of development. And from mesoderm, these tissues arise like muscle, skeleton, kidneys, reproductive system, all the internal organs, they mainly generate from the mesoderm portion. And the endoderm, it gives rise to endocrine glands, lungs, digestive tract, liver. So as you can see that these 3 germ layers are responsible for giving rise to different organs and tissues of our body. So that is why I will just give a brief introduction of these terms, and later when we will do gametogenesis, fertilization, we can talk about it in details. So the first topic that you have in this section, the basic concepts of development, is the cell potency. So what is cell potency? Cell potency is basically the varying ability of stem cells to differentiate into specialized cell types. What is that? Nothing but how the stem cells have different abilities to develop or to differentiate. The term that we are using here is differentiate, because it is not only dividing but it is also getting converted into a specialized cell type. At first it was not that cell type, but after division that cell type is generated. So we call it a specialized cell type, and this process of cell division is known as differentiation, because they are not reproducing another of its kind, but they are totally producing a different cell type. So As we know that stem cells have this ability to give rise to a different type of cell. Now, the ability or the degree of potential that a stem cell has is known as cell potency. So, cell potency will be greater in cells which can generate more types of cells, and the potency will be lower as when the cells cannot generate many cell types, only a few cell types, then the potency will be lower. So, according to that, potency can be classified into the following three types. Major three types are totipotent, pluripotent and multipotent. And some other terms are there like oligopotent, unipotent, nullipotent, which are not very much used, but still it's better if you know what they mean. So according to relative potency, like exactly at which level their potency lies, I have arranged them in decreasing order from highest to lowest. So as you can see, the totipotent cells have the highest level of potency. So what are those cells? Those cells are which can differentiate into any cell type, like any cell type of the body, including the extra embryonic cells or the placental cells. So extra embryonic cells, which is the placental cells, as you know that placenta is the external covering of the embryo. So this we are calling it extra embryonic. So not only do these cells have the ability to differentiate into any cell of the embryo, but also it can differentiate into a cell of the extra embryonic portion, which is the placenta. So toti means whole or total. So that gives explains why this name is given. So for example, the zygote, like I told you, that zygote is the first formed cell of the living organism. So not any living organism, but a multicellular organism. So from zygote, obviously the entire body can be developed. Like it has the ability to give rise to all the cell types that an embryo needs, as well as the extra embryonic cells. So these zygote or the early morula, like I showed you that this is the morula stage. So in early morula stage, like 16 cell stage, 32 cell stage, these cells have the ability to give rise to any cell type. So they are totipotent cells. So from zygote to the morula stage, you can call them totipotent. The next is pluripotent stem cells. So what are pluripotent stem cells? Obviously, as you can understand, their potency is lower than totipotent stem cells. They can differentiate into any cells from the three germ layers, that is all embryonic cells. So what are the three germ layers? The three germ layers are ectoderm, mesoderm and endoderm. So any cell that can differentiate into either ectoderm or mesoderm or endoderm, that cell we can call it pluripotent. So so exactly at which stage does this cell come? Embryonic stem cells, induced pluripotent stem cells. So pluri means many, and from here the name is given. So embryonic stem cells and induced pluripotent stem cells, they are both pluripotent cells. So induced pluripotent stem cells are basically known as iPSC, which is an artificially derived pluripotent cells. But in case of natural cells, you can call the embryonic stem cells are pluripotent because they can give rise to any three germ layers: ectoderm, mesoderm, endoderm. So that covers all the cell types of the embryonic body. I'm not talking about the extra embryonic or the placental cells. They cannot give rise to them. That is the ability of totipotent cells. So next, moving on to multipotent stem cells. Their potency is lower than pluripotent. Multi means several, so they have a limited range of cell types into which it can differentiate. So the hematopoietic stem cells, neural stem cells, mesenchymal stem cells, these are the examples of multipotent stem cells. Why? Because the hematopoietic stem cells are responsible for differentiating into all the blood cell types. So as we know that blood contains a lot of cell types from RBCs to WBCs and platelets, and then WBCs have a different are of different components like the lymphocytes, the monocytes, and then the basophils, eosinophils, neutrophils. So all those cell types can be formed from the hematopoietic stem cells. So that is a limited range, but still it has several cell types. So that means that hematopoietic stem cells can give rise to all the possible blood cell types that we have. Moving on to oligopotent stem cells. Now they have very low potency level and they can differentiate only into a few cell types. Oligo means few, and their example is lymphoid or myeloid stem cells. So lymphoid stem cells are basically derived from hematopoietic stem cells only. Lymphoid stem cells are responsible for differentiating into the T lymphocytes and the B lymphocytes. So you can see that this is a small subset of stem cells and it derives from the multipotent stem cell only, but they are oligopotent because their potency is lower than them and they can give rise to 2 or 3 cell types. The next is unipotent stem cell, which has the lowest potency in among all these cell types. It can differentiate into only one cell type. So unipotent stem cell example is epidermal stem cell, which is the skin cell basically. So whenever we get a bruise or our skin gets chipped off, then the epidermal stem cell generates new skin cells which cover that portion. So that is an example of unipotent stem cells. And then in case of burn patients also, you will see that some skin grafting is done, like skin from other regions of the body are grafted into the burned portion and then new skin cells develop from there. So that is so the epidermal stem cell is an example of the unipotent. It can give rise to only one cell type, that is the skin cell. Lastly, the nullipotent stem cell, which means it has no potency at all. It is a terminally differentiated cell and they do not go through any cell division nor any differentiation. So null means 0, you all know that, and the example is erythrocyte or an RBC. So it's an obvious thing that RBC, they do not divide. So it is terminally differentiated. They neither go through any cell division and differentiation is just out of question. So in that case, we can call it a nullipotent cell and its potency is none. So I think the cell potency concept is clear to you now. And moving on from here, we will... Okay, so this is basically a diagrammatic representation of what I just said right now. Cell potency from totipotent cell to unipotent cell, the potency decreases. So it starts with a zygote when the oocyte and the sperm is fertilized, and this is all known as a totipotent cell, and from here it after subsequent cell divisions it forms the blastocyst structure. So I told you that blastocyst is then giving rise to ectoderm, mesoderm, endoderm and forming the gastrula. So we can call it a pluripotent cell. And then from ectoderm, mesoderm, endoderm, the different subsets of cells are getting generated. So here we can call it a multipotent stem cell. And after that, when the cells are only differentiating into one cell type, it has become a specific cell type and it cannot further differentiate into any other cell type. We can call it a unipotent cell. So from zygote to unipotent cell, the potency decreases. The more the cell types of the cell of a stem cell which it can differentiate into, the more will be its potency. And the lower however, when it will progress towards development, its potency will decrease.